Hello, I'm Rob Burgess. I'm Chief Business Officer for Sino Biological, and I want to thank you for attending my seminar today titled Key Immuno-Oncology Targets, which I put together for Cambridge Health Tech Institute's 2021 Discovery on Target Conference. During this seminar, I will briefly outline Sino's offering in the Immuno-Oncology Target Research category, and then I'll present a few case studies, and then I'll finally finish up by saying a little bit more about our company, as well as some related product offerings and services offerings that you might be interested in. Since I only have a few minutes, I'm going to go ahead and get started. So Sino Biological has a very comprehensive offering of recombinant proteins, as well as antibodies and even immunoassays, which cover many different immune checkpoint targets. And in fact, we're covering around 80 key immune checkpoint targets with a portfolio of over 400 full-length bioactive recombinant proteins, and then related monoclonals, polyclonals, and recombinant antibodies totaling about 700, which recognize strategically chosen immunocheckpoint proteins. These cover families such as the ICOS family and its corresponding ligands, the PD-1, PD-L1 interaction pathways and corresponding family members, the Siglec family, the TGF-beta and B7 superfamilies, and even a variety of collectives. These have been validated for a variety of different assays, including immunofluorescence and immunohistochemistry, Western blot, and a number of others, and cover species such as human, mouse, rat, and quite a few other species. So Sino indeed has one of the world's largest offerings of immune checkpoint target-related recombinant proteins, antibodies, and even corresponding ELISA kits. Here's some examples of data for antibodies that recognize immune checkpoint targets that have been validated for various applications. In PC3 cells, you can see here that B7H3 is very efficiently detected by an anti-B7H3 antibody by immunofluorescence. And here are two examples of immunohistochemistry for the detection of mouse PD-1 and human BC7 in a mouse kidney tissue slice that's been paraffin embedded, and a human tonsil tissue paraffin embedded tissue slice as well. By flow cytometry, here's an example of detection of B7, also known as CD86 on body cells, using an anti-B7-2 antibody. And in fact, a number of antibodies also show full neutralizing activity against their targets. Here's an example of that, and particularly the effect of an anti-human PD-1 antibody on the expression or release of interferon gamma and IL-2 in mixed lymphocytes. And what you can see here is that in general, as you increase the concentration of the antibody on the treated on the lymphocyte, you see an increase in interferon gamma concentrations, as well as an increase in IL-2 production and secretion. Here are just some examples of validated antibodies which both neutralize and fully block the function of PD-1 and the PD-1 ligand, PD-L1. So the first case study I want to review with you is by Jeremy Waite and colleagues at Agenus. And what Jeremy's group did was they studied the co-engagement of the Fc gamma receptor with its corresponding receptor CTLA-4 on adjacent cells. So the Fc gamma receptor on antigen-producing cells, and then CTLA-4 on T cells. And they studied how this actually modulated the activity and how therapeutic antibodies could disrupt or enhance this activity to reduce tumor size in mice. So the goal here really was to develop an antibody that had been optimized to drive CTLA-4 signaling and its interaction specifically with the Fc gamma receptor on antigen-producing cells to basically reduce tumor size in vivo in mice. They utilized a recombinant human antibody, recombinant protein, CTLA4 his-tag protein available from Sino Biological. This is a Coumacy stain showing the purity of that protein, and they developed a variety of different anti-CTLA4 human antibodies that recognize CTLA4. They then employed surface plasmon resonance to confirm the affinity of these antibodies to actually target and bind very tightly to Sino's CTLA4, and then in conjunction optimize the interaction between CTLA4 and the Fc gamma receptor on antigen-producing cells. So here's a classical study of the effect of CTLA4 and specifically the inhibition of IL-2 secretion. So you can see that when you add the CTLA4 that Sino Biological provided, you get a dose S-curve response suggesting its bioactivity and the inhibition of IL-2 secretion in cells. What they did when they took their final optimized antibody and they injected it into mice, they then studied the proliferative effect of tumor cells in mice using a TCR-Ki67 assay. And you can see that at 3 days, a non-specific antibody does not affect or inhibit the proliferation of tumor cells in mice, but the antibody specific for CTLA4 that was generated using Sino Biological CTLA4 protein did in fact have an extreme dose response at 3 days after injection and fast reduction in tumor cell proliferation in vivo. And then correspondingly, what you can see is a vast reduction in tumor size utilizing the anti-CTLA4 antibody versus, in contrast, a control non-specific antibody. And so what they proved then was that the antibody developed that recognizes CTLA4 provided by Sino Biological indeed enhances the interaction between CTLA4 on T cells and the Fc gamma receptor on antigen-producing cells. And this causes an anti-tumor effect. A very nice study by Jeremy and his colleagues at Burgess utilizing Sino's human CTLA4 his-tag protein. And the second case study is by Yunlong Zhao and colleagues at the University of California, San Diego. Yunlong and colleagues studied the interactions between PD-L1 CD80 on antigen-producing cells and how these specifically interact with T cells. And what is the interplay and the contrast in play between these receptors and interactions with their ligands on T cells. And the way they did this was they actually utilized a variety of proteins provided by Sino Biological. This includes CD80, CD86, PD1, PD-L1, L2, as well as ICAM. And the goal of this study really was to determine how these receptors and their corresponding ligands interact to affect signaling between T cells and antigen-producing cells. And the way they conducted this experiment was they studied T cells in a supported lipid bilayer assay. And they looked at the specific clustering indices and effects between complexes of CD80 and PD-L1 and their corresponding antigens present on adjacent cells. So what Yunlong and colleagues showed was that as you increase the concentration of CD80 in the single supported lipid bilayer assay, you see a disruption in the effect of the PD-1, CD80, CD28 interaction and an enhancement of CD80's interaction with CTLA4 on T cells. As you then carry out an increase to PD-L1, the ligand of PD-1, concentration above that of CD80, you start to see a switch and specifically CD80 and PD-L1 become much more engaged in interacting, and this thereby disrupts the function of the PD-L1 and CTLA-4 interaction between T cells and antigen-producing cells. And they were even able to show that utilizing an anti-PD-L1 antibody provided by Sino Biological, that they could disrupt the interaction between PD-L1 and PD-1 between antigen-producing cells and T cells, and thereby enhance the CD80 CTLA-4 interaction. And their conclusion was that PD-L1 actually heterodimerizes with CD80, as you can see in this graph, to weaken CD80 CTLA-4 interactions, but not CD80 CD28 interactions. Again, a very nice study by Yinlong Zhao and colleagues at UCSD, utilizing a variety of Sino Biological proteins as well as antibodies to look at the interplay between antigen-producing cells and T cells, and specifically the key immune checkpoint targets and the proteins involved in these immune checkpoint pathways. So just a little bit about Sino Biological. I want to mention that our company was founded in 2007. It's been around now about 13 years or so. And it was founded based upon the production of full-length bioactive recombinant proteins as a spin-out specifically of MIT. The company is headquartered in Beijing. We have 110,000 square feet of lab and office space there. And 100% of all products that Sino Biological sells are manufactured, developed, manufactured, and produced in-house in Beijing. The company is now publicly traded in China. We have almost 500 employees, about a third of which have upper-level life sciences-based master's or doctoral degrees. And the company's core business really is focused on catalog research use only or diagnostic products and related CRO services. It really is a one-stop reagent and service provider. And we have now cloned virtually the entire human genome, 22,000 plus cDNAs, into about 20 different types of eukaryotic and mammalian cell optimized expression vectors, to give us a portfolio of about 400,000 eukaryotic and mammalian expression vectors to choose from. And hence, as a result, we have almost 8,000 now full-length bioactive recombinant proteins. And we've chosen the best targets here and generated almost 14,000 recombinant polyclonal or monoclonal antibodies. And we have a variety of finished ELISA kits and other types of immunoassays and thousands of validated antibody pairs as well. And I also want to mention that we have related CRO services for every type of product that you see up here. And really, we can affect every stage of the drug discovery and development pathway, from initial antibody and protein discovery, through optimization of those leads, high throughput characterization of hits related to that, and specifically the bioactivity of those, and ultimately how these affect biological processes. Here's an example, or really a summary of Sino's Immuno-Oncology reagents. Of course, we have immune checkpoint recombinant protein targets. These are listed here, and I just spoke about some of those in the case studies. We also have monoclonal antibodies for cancer immunotherapy, and even vaccine-related antigens for cancer immunotherapy. And a variety of biopharma, and biotech are buying our recombinant protein antigens for the development of their immunotherapies related to vaccine research and development. And then finally, Sino is one of the world's largest providers of cytokines, and specifically for cytokines related to cancer immunotherapy. So I wouldn't be doing the world justice if I didn't briefly mention Sino's COVID-19 product offering. We do have the world's largest selection of COVID-19 related products and reagents. And on average, as a new variant of COVID-19 is announced, we have the protein and the recombinant antibody, spike and nucleocapsid, for example, in our catalogs and launched about 2 weeks after the release of the nucleotide sequences for those new variants. So we have recombinant antigens, of course, spike protein and nucleocapsid proteins. These are come in a variety of forms, tagged and untagged. We also have cross-reacting antibodies. There are some examples of rapid polyclonal antibodies, which bind to the spike protein and the specific subunits of the spike protein. We have COVID-19 ELISA kits, and we even sell the antigen expression vectors that we utilize to generate these most recent variants as well. Just to finish up, Ben, this is a listing of our infectious disease offering, or an overview, and I show this because we know that infections often result in inflammatory responses and key immune responses and have a direct effect on immuno-oncology and immune target activity. Sino actually now has over 1,000 full-length recombinant antigens related to viruses, representing about 350 different viral antigen strains. This is the world's largest selections. The world's largest selection, and all of these are ISO 9001 and 13485 certified. In addition to COVID-19, we carry a variety of other coronavirus-related antigens, and then other viral antigens as well, representing RSV, HCV, hepatitis, and quite a few others. Our COVID-19 related antigen bank now tops over 200 SARS-CoV-2 related antigens, and then roughly 170 SARS-CoV-2 related variants. Many of these have been biotinylated for easy tagging. And then finally, we have over 630 recombinant flu antigens, and these include flu antigens for 2015 through 2022 vaccine strains. So with that, I just want to thank Cambridge Health Tech Institute for inviting me to give this seminar at 2021 Discovery on Target. And I also want to thank all the attendees for joining me for my seminar today. I hope you enjoyed it, and I hope you enjoy the rest of the conference. Thank you very much.